put my boots on and lace them up. Hard work, not another day's work. Hard work, hard work.
So, in preparation for this, I did a little bit of background reading, particularly around interviewing. This is my first appointment as a science librarian. So although I've been in librarianship for many years, I was a business librarian. It's very different than speaking to someone whose papers you cannot understand, and talks in a very different vocabulary. I don't have a science background. I have a humanities background. So this was of particular interest to me. How do I obtain information from Jose, from other faculty members, and also sort of sell my services and try to add value where I can, okay, and try to find a common language. So one of the things I came across when I was researching was this idea of elite interviewing. Has anyone ever heard of this before? Elite, as in elite universities? Elite interviewing. Generally, it is part of when you're going in to interview the president or interview politicians or interview very high-level executives that run multi-billion-dollar corporations. Although Jose is very fabulous, he is not an elite in the terms that they have defined it in those articles. Certainly,
happy.
What?
I'm very happy I'm not.
So, but reading that and coming from a non-scientific background, the power dynamic is kind of like that. Jose has all the information. Jose knows all the stuff, okay? I have a different ability, a different set of knowledge, but I'm trying to get to him to find out what he knows and actually prove to him that I can be helpful to him and not just a pain in the neck saying, Jose, can we talk for a few minutes? Okay, that I can add value to his enterprise. And to do that, the main thing is really doing your research. Okay, and I actually did this again last night. I spent an hour and a half trying to read the articles that are on the lab's webpage. And I got a basic idea of what he does, okay? In addition to that, I've taken some MOOCs and been reading, trying to bring myself up to speed in the life sciences, okay? And throughout the course of the interview, you might start off being in a deficit position, but the hope is to find common ground, okay, and to position oneself as a partner, okay? And it takes a series. It's very difficult to do this in the short time, but it's something that you try to do. Social skills aren't that important, okay? They're important, but it's not the crux of it. The crux of it really is listening. Being able to listen. And most people don't listen to one another. Most people can't wait to get the next word in, okay? They're already thinking about the next thing that they're going to say instead of really thinking about what is this person trying to say to me, okay? Because of that, I went with an unstructured interview, all right? I have a bit of an agenda. He knows I have a bit of an agenda. He knows I want to try to get my services, okay, in helping him. But at the same time, I want it to be a very genuine, organic exchange, and I want to learn from him as much as I can, okay? So careful listening, it involves intense concentration. It's actually work. Okay, I have my notebook that's full of my notes from different conversations that I've had with other faculty. We're gonna do this. We haven't done this official interview thing. We're gonna do it in front of all of you. I'm a little nervous about it, but Jose is very tactful and wonderful, and I feel pretty confident about getting to the crux of things. So part of becoming a good interviewer and an interviewer of researchers is to see yourself, okay? Just like teaching, how many people have videotaped themselves teaching? Yeah. How many people were horrified by what they saw? Yeah, exactly, okay? This is sort of the same thing. I tend to be a little, like, kind of, I fear that I get into, like sort of the female nurturing, oh, really, isn't that nice? Oh, tell me about that. Sort of that kind of thing. And you can tell me afterwards if I fall into that, okay? What I want to have really is a genuine exchange between two minds. And I don't want to come across as the helpful librarian, okay? I want to come across as a professional librarian who's competent and capable and intelligent. So a lot of that is in how you dress, how you hold yourself, how you speak. And I think it's really important to see yourself being yourself and have that objectivity. But there's also has to be some genuineness to it. For me, that's people can tell immediately. You can tell if I was not genuine, okay? I'd rather tell someone I don't know what I'm talking about than get up there and blah, 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 because he's going to know immediately, right? Yeah, I'm not, yeah, he's pretty smart, okay? So being genuine, being aware of my own cognitive frames, what I'm bringing into the conversation, okay? The fact that I don't have the education that he has, I don't have the background he has, okay? I don't have the international fabulousness that you have, all that sort of thing.
What I would add is, when I teach my graduate students, my postdocs, you always have to know who is the other person that is on the other side. And she already mentioned who is the other person in terms of research and those papers. But who is the other person as a person is also important. I'm interviewing a just starting faculty, 35 years old. I am interviewing a retiring faculty, 65. I am interviewing a guy that has a lab of 10 people. I am interviewing a guy that, well, just teach a couple of classes, one graduate student, because what they have in mind about that interview is going to be very, very different, right? So if you have a lab of 10 people and you need to have 2 or 3 grants, every minute counts. So when she walks in, is what do you want? What is in it for me? Mm-hmm. It's a very business-oriented, as I think she was mentioning. It's an enterprise. So
yeah. I would just also like to say that the first time we met, Jose emailed me prior to our meeting and said, I have a half an hour to meet with you. We had an hour scheduled. And that day, an hour and a half later, after he's taken me through the labs and introduced me to his graduate students, and we've had an amazing conversation about where he wants to go and where he's been, both in a professional manner and in a personal manner. We've developed a congenial relationship that will allow us to work.
But I didn't know you, so the default is very short.
Yes, of course.
Just not to be impolite.
Of course.
Right?
Of course, right? Half an hour. I came in and he was like, "Oh, okay, let's go." All right, so we're going to start the interview. We're going to maybe pretend that we're sitting in his office, even though this is not how it would work. So that's why I have my work outfit on. So, Jose, hello.
Hey, how you doing?
I'm good. How are you doing? Nice to meet you.
Nice to meet you too.
I'm Rachel. I'm with the library.
Okay.
And I would like to discuss your research today. I read a little bit about what you do, and it has to do with biometals and membranes and the transport of such biometals. Could you tell me a little bit more about that?
Sure. Cells that may be your cells, my cells, the cells of the plant, the bacteria that is floating all around. Basically, it's a bag, right? Have a membrane around
Yeah.
that define the inside and the outside.
Okay.
So then you have these metals. What are the metals? We know that we need these things like iron because it's in the blood, and zinc and copper is good for us. We have to get that, and we get it with the food. So the question is, once that is out there, right, and we put it in how it goes inside the cell, how it goes across the membrane, okay?
Okay.
It just cannot diffuse into it, okay? It cannot do that.
Is that because it's hard?
It's because they are ions and they have charges, right? And the membrane is made of lipids. And the lipids don't like water and ions.
Lipids are fat, right?
Indeed.
Yeah. Know all about the lipids. Okay, great.
So, in particular, we study in bacteria, right? Because bacteria are much more simple, easy. And then we started there many years ago, and then we started moving, okay, and this is something that developed in the last 3-4 years, this idea that these metals are at the center of the bacteria interaction with, the bacteria interaction with the host, the infection process
Yeah.
of a bacteria with the host. The metals it's a battle between the host and the bacteria.
Yes.
They both try to kill each other,
Yes.
and they throw at each other these metals
They throw the
Yes.
metals at each other.
because they are toxic too. So we expose the bacteria to, for instance, a lot of copper.
Okay.
We do that. And the bacteria, what it does is take the copper out because it doesn't want to get hurt.
But it needs some of that copper.
Needs
at the
a little
same
bit.
time.
Right. That's what is called homeostasis. To have the right amount, not more, not less. And for that, are all these molecules that we study, these proteins.
So you would take some of these biometals
Yeah.
in your lab under lab conditions and infect.
Now, what you do is you take the bacteria,
Yeah.
correct? And you take out the gene that move the metals. So now you put the bacteria that cannot move the metals.
Wait a minute. The gene moves the metals.
The gene makes a protein,
The gene makes the protein.
and the
that
protein moves the
metals.
metals.
That's right.
So you take it out, so the bacteria cannot move the metal anymore.
Okay.
Now, can the bacteria infect or not? That's the question.
I would say it cannot fight.
Right, it cannot fight, it die, we win.
You win. I thought we were for the living being.
You are in favor of the bacteria now? No.
I don't
no.
want to be in favor of the bacteria.
Right, so if the bacteria is lacking the gene, cannot infect. So the host is not sick.
Oh, the host wins because you're taking the gene out of the bacteria.
Exactly.
So the bacteria can't throw the
Cannot
the
defend
metals.
itself. Right?
Okay,
So that's what we do.
it's kind of important. Now,
Yeah.
how bacteria is everywhere, right? It's ubiquitous.
Yeah.
We know that. I'm wondering, clinically wise,
Yeah.
what would be the next step? Would you go in if how do you like control the
this is-
bacteria
yeah,
of the world,
yeah.
right?
No, I understand. So, and here come a little bit, I don't care.
Okay, you've won the fight.
No. I don't care because I'm not very interested in applied science.
Okay, Okay.
Really, I don't want to cure anybody. It's a radical concept, isn't it? But some of us just do science for the fun of it, to have new knowledge, correct? And this is a very interesting process. Now, I also need to obtain funding from NIH, so I have to explain to these guys how you're going to cure somebody. So if I identify one of these proteins that is very important, right, and if I can annulate, repress it, remove it, the bacteria dies, that becomes a very nice target for drugs development.
Yes.
So you can design, so you identify the target. This protein is important for the survival of the bacteria in the host. So you make a drug that inhibit this protein, inhibit this process, the bacteria dies. So that's why that could be an application.
What are some of these proteins that you've identified?
So they are called ATPases. They hydrolyze ATP. ATP is this energy money in the cell, and they hydrolyze that, and basically they function like pumps. They pump the iron out. So the energy to pump the iron is this ATP. So they are metal transporting ATPases, correct?
Okay.
So, for instance, the thing that we have published in the last year is we identify one that transported an ion that's called manganese.
Manganese?
Manganese, and is relevant for tuberculosis survival. If we take it out of the tuberculosis or we inhibit, that tuberculosis cannot infect an organism.
Okay.
So that's one that we identified recently.
Okay. I have lots of questions. How do you identify, do you just follow the copper? Do you follow the zinc? Do you follow
No.
How do you
No.
identify the bacteria that are associated with?
Okay, so what you do is reverse genetics. So basically now those organisms are all sequenced, right?
Yes.
Have the DNA sequence and
Yes.
it's databases.
And that's
With-
in like NCBI.
Exactly.
Okay.
So we do data mining for proteins that we suspect are going to carry ions. Okay.
Oh.
Because these proteins are like, I don't know, if you go around and you find something this high with two wheels, it's a bike, right? That's more or
Yeah.
less the same thing.
Okay.
You look for parts of
Okay.
that pump,
Okay.
right? If you go out there and you find something with two wings, it's a plane.
It's
That's the same principle. So you can data mine and identify these proteins, and you say, oh, this likely does this job, which is to move the ion. So then you have the sequence, so you can clone it, take it out from the bacteria, make it, and see the function. See if it's indeed a transporter of manganese or whatever.
Well, it's interesting that you mentioned the data mining because I just back in April went to NCBI for a week, NIH, which I'm sure you're familiar with, to learn how to run some of those databases, to do data mining, to perform some bioinformatics in just this sort of application. Who do you have now, which in your lab that does that work for you? Is it grad
Okay.
students? Do you do it?
Well, there are two tasks that go into this identifying the target. Just identify the target is usually me. I fool around in the thing and I see something that look weird and I can pick it up. That is just me. It's something that you can even do for a distraction. But once that you have the target, now you have to do blast searches. You know what blast?
I know blast. Yep.
Okay, blast searches, and you start looking. Okay, where is this protein? Where I find a lot of them, in which kind of organisms. All that is done usually as part of a project by a graduate student,
Graduate students. Okay.
or maybe a postdoc. And usually it's a graduate student, and then somebody else have to do it again because they are not very good at it.
Okay.
And from time to time, this is interesting. From time to time, we have this senior thesis.
Yes.
So this is a frequent problem. You have these undergraduates that are brilliant, hard worker, terrific people. But some of them, like all of us, don't have skills in everything. So there is always the graduate student that can think, can write, can do everything, but cannot work with his or her hands, right? Those individuals exist, right? So what do you do with that person? You have to do a senior project. You give him or her a bioinformatic project.
Okay.
So that is very common. So that is, and sometimes you're going to, for instance, right now I have an undergraduate student that we wanted to do a bioinformatic project together with some people from Mexico. And so I sent the undergraduate to spend a summer in Mexico to do all the bioinformatic. And so she learned, and now she's writing a paper and graduating. But the thing is, we do that all the time, and depend. You have to match the activity with the person.
Which leads into your international collaborations. I've been going through your publications. You've got people across all different oceans that you're working with. I'm wondering, how do you share information with them? How do you share your data sets? How do you share your...
Dropbox and Skype.
All right. Dropbox and Skype.
Yeah.
Are you using WPI's like research at WPI?
We were
Our
just talking. I
infrastructure?
don't know where she is now.
There she
So
is.
we were just talking there. That have to be optimized. It's
Okay.
too slow.
It's too slow for you.
We are using it, but it's too slow.
Okay.
And that has another problem.
Okay.
It's accessible to WPI employees, not to my collaborators.
Yes.
And that's okay, not a problem. But I have other media to communicate with them.
Okay.
That is the Dropbox. That's
Okay.
I
I know those guys that are working on that, Sierra and Rafaela.
Yeah.
Do you know Sierra and
Yeah, yeah.
Rafaela?
I
Okay.
know.
I know that they're trying to work on a solution for that, but it's getting stopped somewhere. Would it be possible, since we're running out of time,
Yeah.
would it be possible to have a meeting with Sia and/or Rafaela, myself, you, and maybe one of your graduate students or the whole lab to come in and talk about these things?
Too much talking. Right. No, seriously.
Okay.
Besides, I already talked. Raphael is Italian.
Yes.
He's talked too much.
He does talk a lot.
I don't have time to talk with him for half an hour. It's very simple. If it's fast, I use it.
Okay.
It's not fast, I don't care.
Okay.
I move on into something else.
Okay.
I'm exaggerating. These are two fantastic guys, right? But I'm exaggerating just to provide that kind of thing. It's not my job to go to evade the solution. It's their job to provide me the solution. And again, but it may sound a little bit arrogant, and it's probably arrogant, but that's okay. The thing is, I am bringing overhead. I am paying for his service, right? That is critical when you talk with your faculty we all come from different schools, right? Some small state school, small colleges, some research universities. Again, the person you are talking is very different. So if you are in a research university talking with a guy that is bringing, I don't know, $250,000 a year in overhead, that guy wants service, right? It's a
Like a business,
It's a business. So that,
right?
yeah. Sorry, I have a question over there.
Oh, yeah, okay.
But that guy has to understand what your problem is. So, I mean, maybe.
No, well, yeah, of course. Yeah. No. Yes. No, that's reasonable. Yeah. But they do know that this is love.
Yeah, they know what the problem is. They know what the problem is.
Yeah.
That was a bad question or a bad lead-in on my part. I should have grabbed onto the bioinformatics thing and the grad students instead of trying to push for
So-
the data management stuff.
Let's
My bad.
let's explore the bioinformatics.
It is.
Or no, let's explore it.
Oh, let's explore it?
Yeah.
Okay.
So my question is, what can you do for me with the bioinformatics?
I can blast. I know
You
how
can
to
blast.
blast.
You can do
Yes.
sequence alignment.
I can.
That's
I know how
good.
to do all of it. Yes. I'm not.
No. You don't.
I know how to do it.
Or you know how to train people to do that.
No, I know how to do it. I don't know
But
how
that
to
go-
pull the sequencing out of the thing that you use, but if you give me the sequence, I can blast it.
So, I was talking this earlier that I don't know what is your job description.
Okay.
Correct? So I may ask questions like, now, we get all the information, right? And then we have to do homology trees.
Yes.
Can you give me good software to do that?
I don't know, but I'd have to investigate. I'd have to research the homology software. I don't know off the top of my head,
Fantastic.
but I can certainly look into it for you.
Okay. So that is the kind of thing I'm looking, okay, what she can give me. Can she solve the problem of, because another problem I have is that people rotate in the lab, right? It's not like we in a library, we work together for 15 years with the same person. My people leave every a postdoc maybe 2-3 years, and that, a postdoc, it could be very good if you understand what is the role in each lab. In a lab, the role of the people. The PI is me, I have to write the grants, bring money. I am always in somewhere else, right? Then maybe a lab manager that may have a PhD or not. That person have been there with that boss for a long time, have history. I don't have that. Then come the postdoc. The postdoc is a gun for hire. That person is there to get data and papers. Very fast. There is no time for learning. The person cannot produce data in 3-4 months. Goodbye. That's the nature of most of them are foreigners, right? That kind of thing. Graduate student is going to be in the lab for 5 years. He or she needs time to learn. We don't expect that they do anything in the first 2-3 years. The maximum productivity is going to be in year 4 or 5. But then, with these people rotating, the lab have very little memory. They have memory in terms that the data is archived, it's in the computer. But, where I find whatever, that memory is poor. And there is where the people like a librarian, at least in terms of the bioinformatics, or the informatics in general, to make it more global, may be very helpful.
Good. Good. So let's have another time with the graduate students, with or without you
Right.
there, and we can talk bioinformatics and blasting and try to set up some sort of network.
Right,
Yeah,
for working
between
plan.
our offices.
That sounds good.
Okay, great.
That's useful.
Okay, thank you. Thank you
There
for
you
your
go.
time. So
Shall
how
we
did I
ask-
do? Was it good? Was it Was I too smarmy? I hate being smarmy.
I really like how well you communicated the nature of the personnel in the lab, the time, the life cycle of who's in there, and the value of your time. I mean, I know faculty and research time is valuable, but you articulated why. And so now that helps me understand how I can best, if they're in the lab for different purposes, a term paper or a dissertation, it affects what I can
Yes.
help with.
Yes.
Yeah.
Rachel, I like how you were clear about what you knew and what you didn't know. So you weren't pretending to have knowledge that you didn't have, and when you needed him to explain something, you made that clear to him, and then he could explain it to you. That was good.
Could you, Rachel, could you possibly just kind of restate what they're saying, just for context, for afterwards?
So
So,
Oh, yes.
maybe just pull that back, and I don't even know how you would say it, how you'd rephrase it, because it's not really a question.
Okay. Well, she liked the fact that I was able to say, I don't know what you mean by that, or could you explain more of that? And not feel embarrassed by that, because what he's talking about is a highly technical, even though they're throwing the copper, you know, and he was able to put it into those terms for me. And this woman here said that she liked that Jose explained the different roles within the lab, and that it's not a one-size-fits-all solution. Tracy?
Well, so I would like to ask Jose to explain a little bit to us how to get a faculty member to agree to give us that 5 minutes to start talking about what we do. We were talking about this a little bit earlier, that one of the things that we have to, barriers or questions that we need to figure out how to do is to let faculty know what we do and what we can do with and for you. So do you have tips?
well, I guess that different faculty needs different things, and they have more or less open doors, so it's different. In my case,
Jose, sorry, time out. Can you maybe let Rachel just restate
oh,
that?
sure.
Tracy wants to know how we can, if Jose has any tips for how to engage faculty.
It's the same thing that I tell the advisees when they want to come to me, what I tell colleagues to do when they want to talk with the program director at NIH or NSF. Send a short email, not more than a paragraph, with the 3 questions you want to ask and how long it's going to take and how I'm going to benefit. So the meeting is quite defined. It's not an open, let's chat. Sorry.
Okay, we have to wrap this up. Jose will be around for talking
Yeah,
and for
there were
lunch and
people-
that sort of thing.